Hi. Thank you very much, everyone, for coming. We have the company FOTOGLASS. FOTOGLASS is a bio-optical company whose starting point was the synergy between the optics group at Universidad de Cantabria and a large company specializing in decorative mosaics. Its main objective is the solution of problems of different nature through the use of light. It began by developing photoluminescence projects in glass mosaics, continued with the colorimetry studies to determine the aging of wood, and developing intelligent textiles that, thanks to the presence of microparticles, have long-lasting photoluminescence properties in the absence of light stimulus. Thanks to the incorporation of a biomedical research group from the Hospital Universitario Marqués de Valdecilla, this company is starting to develop nanoplasmonic biosensors for the differentiation of tumor versus non-tumor tissues, specifically in glioblastoma, a type of brain tumor. In summary, FOTOGLASS demonstrates how companies can evolve, change, and improve by integrating personnel with a diverse background and expertise, achieving a multidisciplinary group that contributes with new ideas. In this talk, we will have the participation of 3 representatives of the company. First, we have Professor Francisco González, Scientific Advisor of FOTOGLASS and Professor of Optics at Universidad de Cantabria. He will talk about the beginning of the company and the projects that arrive to the market or are arriving to the market at this moment. He will offer a vision of FOTOGLASS evolution. So, Francisco, please.
Thank you. Well, hello, everybody. First of all, we would like to thank the Cátedra SAMCA de Nanotecnología for inviting us to introduce the company FOTOGLASS in this master. And also, I would like to thank Marta and Maria, or Maria and Marta, to help us to organize this session. My name is Francisco González. I'm here with my 2 colleagues, Fernando and José Luis, who are members of the advisory board of the company. And at the beginning, so I'm going to present the main project since 2009. And afterwards, Fernando and José Luis will focus on R&D that is currently being developed using nanotechnology applied to bio field. FOTOGLASS has its origin in the transfer reform made from the optics group to the optics group of the University of Cantabria. We have had high vocation of transference, and in 2008, a company is valid. This factory fabricating vitreous materials of the core demands a solution of photoluminescent problem in vitreous tiles. The solution of this problem was the result of the solution was the foundation of FOTOGLASS as a spin-off centered in the development of photoluminescent materials and color control. In 2009, we were awarded by Sodercan, this institution from the government, Cantabria's government. And in 2015, we are in touching with people from the Hospital Marqués de Valdecilla, working in biotech. And finally, we in 2016, we expand its the specialization of FOTOGLASS to this area, and well, the name is where the life is and light meet. This is the summary, more or less, the talk. I'm talking about the main projects, and Fernando and José Luis, we will focus on things related with the research and development center in nanotechnology and cancer. The first project we developed was around photoluminescent vitreous materials within the framework of construction. Photoluminescent vitreous material is achieved with a sintering process at 700 degrees centigrade of a mixture of ground glass powder and photoluminescent pigments. The final product is tails extremely resistant to weather conditions, fire and abrasion, and therefore ideal for many applications, mainly in signaling, for instance, in subways, airports, tunnels, ports, etc., and also for decor, because it's amazing the effects that it can produce. This product is in the market and is called Starlight. Another application has been in the textile sector, specifically the development of modified fabric to achieve luminescence in situations of darkness. To carry out this product, particles with core cell structure with the core of a photoluminescent material have been used within the fabric. How does it works? Well, it absorbs light, re-emits the light, glows in the dark, and the color could be different, but you can choose the color, but mainly yellow or green. It has many applications. These products have many applications in different fields, other sports, road safety, industry, and the clearest example is that of a firefighter lost in a forest in the course of a fire, and the clothes that the firefighter wears allows to find where he is. This product is also in the market, and the name is Wonders. Oh, sorry. Another sector was the goods sector. Another application was in the goods sector. The problem we addressed here was to define a procedure for matching color in hardwoods. This problem is very difficult because the raw material is very inhomogeneous, and things in different planks of wood coming from different trees, from different places, and etc. So the question here was quite difficult, and from the physical and analyzing of studying measuring the physical properties of this raw material, from the colorimetric properties of the pigments of the paintings, a colorimetric algorithm was developed based on deep learning to achieve color matching. Here, well, these are samples in the lab, and here, this is, I think, is worth to mention this. This is the floor in the Botins museum in Cantabria. This picture corresponds to the floor of this museum. And aspects with the aging of wood by solar radiation and how to avoid it were also addressed in this project. As you can see, in the first stage, the field of operation was optical solutions applied to inert materials. By later, in our second stage of business, we had the opportunity to start working with people belonging to the Hospital Marqués de Valdecilla, giving rise to the creation of a specific line of bio-optics, that is, optical solutions applied to living environments. One of the projects we were involved with, but recently when we are still involved in this, is the application of UVC like germicidal. This line was created because the arrival of SARS-CoV-2 and the new value of UVC and its great germicidal power. By one hand, the appearance of devices using UV lamps in the market without any control of design, of the power, and of the certification about the germicidal effect to eliminate this virus. And on the other hand, the knowledge we had in photoglass in the control of radiation and in the biofield encouraged us to offer our services to lamp manufacturers and customers who needed the use of UVC for disinfection, and such as hospitals, school, transport, etc. And our services has been in this field focused on design devices, bio-optical project, validation and certification, germicidal maintenance, etc. As an example of the kind of work we carry out in this frame, I show the stages of a bio-optics project. We carry out a microbiological study, a radiometric analysis, and a biodosimetric analysis as well. In the microbial study, the main task is to get information about a general microbiological condition of a surface, equipment, or environment. Here, I show an example in a place, in a specific, well, in a hall of a hotel or in another similar place, in which we can detect the hot spots. We call the spots, we can identify the hot spots, which are the points with the highest risk of contagion by contact. Once we have detected this, we have made this, we carry out this task, we proceed to make a radiometric analysis by using a powerful software. And in this way, FOTOGLASS provides all the radiometric studies necessary to define the particularities of the optical system and or test the performance of each manufacturer's products. Here, in this slide, we see a lamp and we see the radiation in this room, and even we can see the shadow areas. This is another example. This is in a bus, in a train, and here we can see, depending on the position of the lamps, the effect of the doses that the different surfaces receive from the lamps. And I have to say that this is also applied to we can not only analyze surfaces, we can only pay attention to our air pipelines. That is very interesting, just to clean the air in a room where the air conditioner is used. And the final step is to do or to carry out a biodosimetric analysis, and this is very useful because this allows to validate the effectiveness of the doses of the UVC radiation. We made a biodosimetry test carrying out using biodosimeters, like this is shown here, that allow the direct measurement of biological material. I have to say here that these dosimeters are homemade, so we constructed these dosimeters, and to do the analysis or to do the experiment, we use non-pathogenical bacteria like shown here, or even ARN of COVID-19. Here we show a picture in which we have the bacteria located in the biodosimeter, and this part of this biodosimeter has not received a big amount of radiation, large amount of radiation. Here, the doses has increased, and here the radiation, the doses has been very high. So, and we can see that here the bacteria has disappeared almost completely. In short, this is well, I have to say, sorry, these projects have applications in many sectors like health, transport, culture, food industry, education, and I have to say that this at this moment is the main source of money of the company, this area. So in short, this is FOTOGLASS, a company focused on using light as a tool in living environments where life and light meet. And given the forum we are addressing today, I give way to Fernando Moreno and Jose Luis Fernandez Luna, who are going to take you on a walk around the R&D of FOTOGLASS. Thank you.
Thank you very much, Francisco. Very nice. As agreed, we leave the questions for the end of the talk. And now, if you don't mind, please stop sharing the
I think it's better to see all the talk.
Yeah. So it's what I was suggesting, that we leave the questions to the end of the talk. Now we move to the next speaker.
Okay.
Fernando.
Thank you. All right. Thank you.
Excellent. So our next speaker is Professor Fernando Moreno. He is also scientific advisor at FOTOGLASS and professor of optics at Universidad de Cantabria. And he will talk about FOTOGLASS Research and Development Department, focusing on nanoplasmonic applications. He will explain the origin and development of the biosensor. Okay, so Fernando, the floor is yours.
Okay, thank you very much for your kind introduction. I would like to say thanks to the organizers of this event and all the institutions involved in this event, like Cátedra SAMCA and the INMA in Zaragoza. Okay, so Paco has told you about our main sources of current main sources of our company, and our idea is to give you or to talk to you about our present, our future, about our recent activities in research and development. In my case, I would like to talk about nanoplasmonics because this is the part where I feel more comfortable, and Jose Luis will talk about the application of these to real problems in biosensing. Okay, so first let me tell you how we become bio. This was not a formal decision, but as in many other cases, a bunch of random situations. In this case, two independent situations met at the same time. One was related to the dissemination of our work in press and social networks, because we were very interested in doing so. And at that time, Jose Luis read some information we published in press about our research activities, the activities of our group. And at the same time, we had a collaborative research or collaboration with an external institution, in this case, Techniker, which is a technological center located in the Basque Country, which were interested in analyzing the plasmonics of nanohole metallic films. So these two, in principle, random situations meet at the same time, and with the proposal made by our colleague, which was, are you able to differentiate between tumor cells and healthy cells with your system? That was the question. And we responded, okay, let's try. And this started our adventure in biotechnology. Let me tell you something about some technical aspects, which I am sure most of you are aware of this, especially my colleague, Luis Martin Moreno. I think he's listening to me, and he's an expert in this area, which is about plasmons and plasmonics, which are the responsible of these applications. People who knows about these effects know that when electromagnetic radiation hits a metal, its free electrons start to oscillate at the incident frequency as an coherent ensemble. And for some characteristic values of the incident frequency, this oscillation resonate, produce a resonance, leading to two important effects. One is for bounded regions like nanoparticles, the radiation can be localized in dimensions much smaller than the incident wavelength, which is in principle surprising result. And on the other hand, you can get a strong field enhancement close to the metal surface, which is the responsible of most of the applications in biosensing. Okay, in particular, let me talk about an interesting effect, an interesting situation, which is when we drill nanoholes in a metallic thin film. More than 50 years ago, a researcher, Bette, published an article in Physical Review dealing with the problem of diffraction by holes much smaller than the incident wavelength. And he gave some results about this. As I said, more than 50 years later, a colleague, Evesen, and his co-workers published a surprising result saying that a part of the results obtained by Bette, they were able to detect some enhancement of the transmitted intensity, but these nanoholes, when we had a periodic structure of these holes in a thin metallic film. This phenomenon was called extraordinary optical transmission, and the researchers making basic research on this effect, like Luis, for instance, got the conclusion that plasmons were the responsible of this effect. This leads to an electromagnetic enhancement just above the metallic surface, which is very, very sensitive to changes in the optical properties of the medium touching the surface. This enhancement produced in the spectrum of the transmitted intensity these peaks, which are very sensitive to changes in the refractive index of the things touching the surface. Okay, the immediate thing is to think what we can apply this to detect small amounts of bio things in fluids. Why? Well, it's very easy to align because the chip is very, very small, and you can illuminate it at almost perpendicular incidence, and you can detect in the same direction the transmitted intensity. It's quite robust. It can be complemented with microfluidic devices in order to guide the liquid problem through the sensing area. Can be miniaturized, which permits the multiplexing of the system. You can detect several, you can do several experiments at the same time, or some detection at the same time, and it presents a very high sensitivity. So in order to get these advantages, you have to choose very well the sensing chip geometry. And in general, it has to be made of a metal which has to be biocompatible, usually it's gold. With high sensitivity, you have to design the diameter of the nanoholes, the thickness of the film, the period, etc., of the parameters in order to get the high sensitivity or high sensitivity in the spectral range you are interested in. The geometry must be compatible with the selected nanofabrication technique. Usually is non-impinged lithography in order to connect with some industrial application. And in general, it should be insensitive to polarization. In this case, we are not interested in polarization effects. So how can we apply this to our problem? In order to solve the question by Jose Luis, can we differentiate cancer cells from healthy cells? So our main hypothesis was that healthy cells present different optical properties, different refractive index in general to those of malignant ones. Also, the structure of a single cell penetrates in the plasmon field of the order of 200 nanometers, 300 nanometers. And most of this penetration corresponds to the membrane of the cell and a part of what we call actin, which is a part of the cytoskeleton. If we choose the sensitivity of the nanohole array given by this equation, which is the ratio between the shift in wavelength of the transmitted peak divided by the change in the refractive index, we applying just a simple effective refractive index model, we can get the value of this shift. Based on this, we made a detailed study through numerical method based on solving Maxwell equation, and we had many different effects. For instance, the flattening effect suffered by the cells. We also included the structure of the cell. We included most of these parameters in our study, and we obtained some result to guide the experiment. So at present, this is our current experimental model. The main characteristic of our system is that we can control the spot size of the illuminating beam on the sensing surface, which permit us to isolate just a single cell and analyze just the optical response of a single cell located on the substrate, in such a way that when the cell is healthy, this produces a peak like this ring, and when this is malignant, this produces a slight shift of the peak, and we can, in principle, distinguish both. So this is the point where we are now. This is the next steps. Now we are trying to develop a system based on all this basic research I have described before, in order to develop an instrument to put in the hands of the doctor in the room of the operation, in order to distinguish, to be able to distinguish between the healthy areas and the non-healthy ones. This is our situation in this moment, and we are trying to develop this kind of instrumental for practical applications. And that's more or less my contribution to this part, and I give way to Jose Luis. I think he can illustrate in more detail these sort of applications, because he is the part of the clinical and the hospital part of our company.
Thank you very much, Fernando. Impressive work. Okay, so we move to our next speaker. We have Dr. José Luis Fernandez Luna. He's also scientific advisor at FOTOGLASS and coordinator of the genetic service at Hospital Universitario Marqués de Valdecilla. He will explain the biomedical applications of the biosensor we have just seen, focusing on cancer detection. So, José Luis.
¿Lo veis? ¿Veis la presentación?
No, not yet. No, it's coming. Perfect.
¿Ahora?
Yeah, perfect. The floor is yours.
Okay. Well, thank you to the organizers again. I'm going to talk to you about the biomedical applications of the biosensor that Fernando just presented.
Try with the prueba con el ratón igual.
Okay.
Perfect.
So the first application I want to talk to you about is the label-free identification of live cancer cells. And why is this important? If we are able to identify a tumor cell and preserve the integrity of a tumor cell, we'll be able to do a number of studies, biochemical, functional, and in vitro studies. And for example, trying to analyze or discover new biomarkers for a better diagnosis, or even to test the different chemotherapeutic drugs in tumor cells isolated from a patient. So we can choose the best treatment for every patient, which is called personalized medicine, which is the medicine of the future. Another interesting application is the identification of circulating tumor cells. Once a tumor becomes metastatic, they release the cancer cells to the bloodstream. So we are able to identify these tumor cells in the blood. We can tell that a metastatic process is taking place in this patient, even before an image technique can tell you that there is a tumor anyway in any part in the body. So the gold standard for cell identification is flow cytometry. And flow cytometry is mainly based on the use of fluorescent labels. These fluorescent labels recognize specific molecules in tumor cells. The problem is that in many cases, we don't know these specific molecules in tumor cells, so we can't use fluorescent labels. And in the absence of fluorescence, flow cytometry can analyze forward and side scatter light from cells. But in this case, the discrimination capacity of flow cytometry decreases. And you can see here on the right, this plot, you can see different cellular populations, and there is an overlapping between all population, including tumor cells and normal cells, like fibroblasts and lymphocytes. So this is not a good system to identify tumor cells in the absence of fluorescence. But before we move on, let's take a look at the complexity of a cell. You can see here that there is a lot of stuff inside the cell. I mean, there are a lot of organelles and structures, like the nucleus, which contains genetic material, but also mitochondria, which are needed to obtain energy for biochemical and metabolic reactions. Also, the endoplasmic reticulum needed to synthesize proteins, or the Golgi system, the Golgi apparatus, that releases molecules which are synthesized by the cell. But also a cell has a skin, which is called the plasma membrane, and a skeleton. And this skeleton is formed by microfilaments and microtubules. And some of these microfilaments are just underneath the plasma membrane. It's called the actin cortex, and is needed to maintain the shape of the cell, and is also needed to change the shape of the cell. So, keeping this scenario in mind, what part of the cell is detecting the biosensor? This is the cell membrane, which is labeled with fluorescence in red, and this is the actin cortex, also using fluorescence. And you can see that this actin cortex forms a ring around the cell, just underneath the plasma membrane. The thickness of this actin cortex is between 100 and 500 nanometers. And the highest density of the actin cortex is around 250 nanometers, which is exactly the penetration depth of the surface plasmon, as Fernando just told you, around 200 and 300 nanometers. So what we are looking here is mainly the structure of the actin cortex. And if the detection capability or discrimination capacity of the biosensor relies on the actin cortex, an interesting question is actin cortex, does actin cortex change in cells? And the answer is yes. You have here two processes, two cellular processes, cell division and cell migration. And in both cases, there is an increase in the thickness of actin cortex. In cell division, cells divide, they go through mitosis, and from one cell you get two daughter cells. And in cell migration, you firstly the cell attached to the surface or a substrate, and then there's a migration, and in this case again there's an increase in the thickness of actin cortex in the leading front of the cell. Interestingly, these two cellular processes are key features of tumor cells. Tumor cells don't have control of cell division. They divide without control, and they migrate to colonize other parts of the body to form metastases. So these features characterize tumor cells. So the next question is the biosensor able to discriminate between cancer and normal cells? And to answer this, we do the following experiment. We obtain biopsies from colorectal cancer patients with the collaboration with the surgeons of the hospital. And in this tissue, we can get both tumor cells and normal cells, going through different steps of mechanical disruption of the tissue and enzymatic digestion. Finally, we get individual cells, again, both from the tumor and from the normal tissue. And we can analyze these individual cells with the biosensor. And you can see here that there is a clear-cut difference in the spectral shift between colorectal cancer cells and epithelial cells, which are normal cells. An interesting thing or interesting result that we obtain is that this discrimination capacity of the biosensor is lost in the absence of actin cortex, which was expected in based on the previous results. You see here a native cell with the actin cortex in red and the cell membrane in green, and we can treat these cells with a compound that destroy or make the actin cortex to disappear. Following the treatment, you see that the actin cortex is gone. However, the integrity of the cell is still there. You can see the cell membrane. And this is what the biosensor is seeing. In the native cell, the biosensor is looking at the cell cortex. And without the actin cortex, we have the proteins around the cell, which are a feature of the plasma membrane, but there are no actin cortex, okay? And in this case, this discrimination capacity, this spectral shift, the increase in the spectral shift that we see in the colorectal cancer cell is lost when we remove the actin cortex. So in summary, this biosensor is able to discriminate between individual cancer cells from normal cells in the absence of fluorescence without any treatment. And this biosensor is able to keep the integrity of the cell for future analysis, in vitro analysis, functional studies, and so on. And also, this discrimination capacity relies mainly on the configuration of the actin cortex, which is a key structure to allow tumor cells to divide and to move or to migrate to another places, which is a characteristic of tumor cells. The second application I want to talk to you about is the real-time identification of tumor margins during surgery. And this may help surgeons to take decisions during surgery. And this is particularly interesting in one kind of brain tumor that is called glioblastoma. This is a very aggressive brain tumor. And the first line of treatment is always surgery. We neurosurgeons need to remove as much tumor as possible, but they have to preserve the normal tissue. That's why it's so important to differentiate, to distinguish between normal tissue and the tumor. So in these tumors, we can distinguish three different parts. The necrotic part, which is in the center of the tumor itself, and the surrounding tissue, which is mainly normal tissue, brain normal tissue. So we wanted to know if the biosensor was able to distinguish these three tissues, these three areas of the tumor. And the answer is yes. I mean, you have here magnetic resonance images of the brain with the tumor marked in yellow with this yellow ring. You have the necrotic part, the tumor itself, and the peritumoral area. The green line is pointing to the site where we take the sample for further analysis. And you can see that we can get these differences in the refractive index change between necrotic tissue, tumor tissue, and the peritumoral area. Most importantly, the difference between the tumor and the peritumoral area, because this is marking the tumor margin, which needs to, neurosurgeon goes to the tumor margin and no further. Currently, we are studying the biological basis of these differences. We need to analyze more patients, more samples to get an idea of the specificity and sensitivity of the system, and we don't know now the biological basis of these differences in the spectral shift. The scenario is completely different to the one I talked to you about in individual cells. I mean, if we analyze individual cells, there is nothing around. But in this case, tumor cells are in the context of a tissue with more cells and more structures. So we can't say here that this difference relies on the actin cortex of the cell, because the tissue is much more complex. So we don't know now the biological basis, but we're working on that. And I guess as a conclusion, I guess the take-home message is that adaptability to the market and innovation are main values of a biotech company where advances and changes are constant and very fast. Thank you all, and we'll be glad to take questions now.
Thank you very much. Fantastic talk and very concise, all the three speakers. We have enjoyed nice view of the company and very nice projects. So now, let's see, do we have any questions here in the audience? Do we have anyone asking the... Oh, sorry. No. Do we have anyone there? Okay, so we are bringing the microphone there so you can listen to the question.
Okay.
They are very shy.
Explaining. In fact, is the one that gives us more money to the company related to the decontamination that you read. I don't know about the airports, big places. So the lamp, the device that you have to deploy and use is it's not portable, and it should be something that is already built there. And then
Maria, it's very difficult to understand.
Very difficult, yes.
Understand, very difficult. Maybe the student can go in there in front of the computer and ask the question.
Yes.
That will be better.
Yeah, much better. Yeah, thank you.
Okay.
Sorry for.
Yeah, okay. Yes.
Come here. Oh, no, come so they can see you.
Better we see as that is a real student and we don't have fake students on the audience. Oh, it's not a real student. No. Looks like a student, but it's not.
I am trying first to encourage the students to ask some questions, but okay, I open the floor. So the first one is for Francisco. I am wondering about the device that you are putting in place for this decontamination at the airports or in the big places. I think it's I mean, about the lamp. How is the lamp? It's not a portable device. I imagine that it's already installed in the building, so you have to work. I mean, you have a specific routine for this cleaning, or could you tell us a little bit more about how is it?
Micrófono, Paco, micrófono. Tienes el micrófono off.
You cannot control this.
I'm not sure. You can check.
Something is no.
Pues pásate a mi despacho.
Yes, maybe I can go to the next one, which is related to this bio application with tumor cells.
Pili. This is moving to the other room.
Yeah,
It's just 2 meters from me.
It's just, yeah, it's like if I move to your room.
Ah, I thought you were in different.
Take it easy, patient.
Hi.
Looks very different.
I don't know what's happening in the screen was completely blocked. I don't know why I couldn't change the microphone or was completely blocked.
Yeah.
So we're talking about lamps and cleaning.
Yeah.
Well, we don't fabricate lamps, obviously. We have.
She talks about the routine of measurement, right?
How do you operate? I mean, I assume that you are working with other companies in terms of
Yeah,
the manufacturing,
lamps, yeah.
And your expertise in this is in this deep learning to in the algorithm to measure in a specific places and make the calculate the doses.
Yes, yes. We have, well, this is a radiometry process. We have a very powerful software, and you can choose you can choose the geometry. You can choose the geometry of the geometry of the space, the place in which you want to locate the lamp, right? You can select the shape of the lamp, the power, and into the software gives you the answer about the doses in any place of the surface into the room. That's right. Well, obviously, when you are putting the lamp in the specific place you have selected, you have to be out of the room because you can suffer damage. But this is so there are include normal, all the lamps have include a safety mechanism in such a way that if the lamp detect any thing moving just around, stop the mission.
Understood.
All right.
I prefer to leave it at the end. Yeah.
I don't know. Because I was going to ask about the lamps as well, but I think it's answered with this question. Then I have another question for Francisco. That I was surprised you said that you were using that you have been seeing me all the time. You said that you have phosphorescent materials that you introduce in glass, but then the sintering process goes up to 800 degrees. So I was quite surprised with that.
Yes,
Oven.
Oven. We are well. The factory has oven and the sinterization process is around 700, 800 degrees centigrades.
Yeah, my surprise is what type of material do you use for the phosphorescent material that survives and is still working after this and sintering process?
I don't remember. Inorganic, obviously, are inorganic material for pigments. I don't remember at this moment. But well, so I think the main advantage of this material is that if you compare, so the signals just around you probably, or you are when you were in a metro station or in any place when you exit are plastic so are paintings on plastic or PVC or aluminum, right? But if a fire appears, these signals disappear, right? However, our materials, our tiles keep without damage.
I think that you are asking about the composition of the photoluminescent pigments, right?
Yes.
I think they are in general, we don't know exactly the chemical formula, but they are erbium doped photoluminescent materials, and we encapsulate it in some.
I'm not sure. Para el textil, sí, pero para
for the textile and
for the textile, yes, but
for the textile
for the tails, no.
For the tails, we don't encapsulate.
No. The tails were inorganic.
They are erbium doped phosphorescent pigments. I don't know exactly what is the formula, but this is a pigment you can you can select from the manufacturer. No, it's not nothing very specific, and they keep okay when they are under 800, 900 degrees, but the process is quite slow. As the oven is quite long, is I don't know, it's 15 meters long. The temperature starts to increase, gets a maximum, and decreases very slowly.
Mm-hmm.
And the final result is really, really good. At the beginning, I remember we had a lot of problems.
Yeah.
As usual.
Not preparing it the first time we tried it.
And obviously, our work is to measure the luminescence, the times just emitting. So in order to classify the materials, well, the A or B, it depends of the necessities of the customer, you know.
Mm-hmm. Then I have another question for Fernando, but then Jose Luis answered that, because in his presentation he says that the idea was to differentiate between cancer and normal cells, and I was going to ask if that was possible, but it was properly answered in the next in the next talk. That obviously yes.
Mm-hmm.
So, if possible, I would like to ask Jose Luis, because he commented that in cells that don't have the actin cortex, then the cells cannot be differentiated. And my question is, can this happen in human body? Because I understand that when you take the cells and you isolate during the processes, maybe you can degrade or remove the actin cortex. But the question is, can this happen in body cells and be this interference in your measurement? I don't know if I explained it right.
The actin cortex is always present in cells.
Always. Okay.
Otherwise, the cell collapses. I mean, the actin cortex is like a skeleton.
Mm-hmm.
It's needed for the cell to maintain the shape. But the configuration of this actin cortex is different from cell to cell. For example, the thickness of this actin cortex, or the configuration, the structure, the polymerization of the actin. Actin is a monomer, is a protein that polymerizes in fibers. So it depends on the polymerization, the length of the fibers, of the microfilaments, and the thickness of the actin cortex, which is different from in normal cells to tumor cells. And that makes the difference. This is why we are able to distinguish both types of cells. But if we remove in one, I show you in one experiment, if we remove, and it's very easy, we just treat the cells with a compound that eliminates the actin cortex. If we remove the actin cortex, we eliminate the discrimination capacity of the biosensor. So this discrimination capacity relies mostly on the actin cortex configuration.
Excellent. Yes, Paula. I think you have to come here.
No. Okay. Okay, so does the biosensor works for any tissue or brain and intestine? Or
I think the question is that if you have a cancer,
yeah, for example,
I don't know,
in the
stomach,
lungs,
yeah, lungs,
lung cancer, or in other tissue, does the biosensor work as well?
Because you have shown examples of a brain cancer, colorectal cancer, and the question is, have you tried with the cancers in other tissues? That's the biosensor work as well for stomach, lung cancers.
Well, this would be a next step. I mean, we have focused on the single tumor type. For individual cells, we choose colorectal cancer cells, and for tissues, for the surgical procedures, we choose glioblastoma. But once we prove that this biosensor is effective in these two systems, we can try to extend to other tumors. I mean, we think we can, at least in cells, because in individual cells, we know the biological basis. We know that this discrimination capacity relies on the actin cortex. We don't know about the biological basis of the discrimination capacity of the biosensor in tissues. As I told you, tissue is much more complex, and we are now studying on this. But we think that we are able to extend these capacities of the biosensor to other types of tumors. But again, we have to prove it.
So, in the idea, sorry, Jose Luis, just to add something to your comment, the idea to focus on glioblastoma is just because the surgeon, the neurological surgeon, has the problem of differentiate between tissue, healthy tissue, and cancer tissue, and we focus on that cancer in order. And our idea is to develop an instrument based on our basic research, and an instrument he or she can handle in his or her hand in order to touch in different parts of the brain in real time, just in order to differentiate that. That's one of the selections of this kind of tumor in the case of the brain.
I mean, tumor borders are always a problem for surgeons, but in some cases, for example, in brain tumors, this is critical, because you want to remove the tumor, but you want to allow or preserve the normal tissue. In other tissues, for example, in colon, in intestine, is not so critical. You can remove a little more of the tissue and nothing happened. But in the brain is not the same situation. So I have to be very accurate in removing just the tumor and not anything else.
That's right. And that was the idea of my last transparency.
Okay, I have my hand on.
Yes.
Go ahead.
Okay, so thank you very much for the presentations. I really enjoy, and I think the company is a very good combination, and you have many different fields of.
Ooh.
We lost her.
Yeah, we lost her.
Finish the Wi-Fi in the office.
Yeah.
Yes.
Maybe it's in the
She will rejoin.
on the mountains, probably. So behind her, there's a high mountains, or is has a problem with the signal.
Yeah, with the connection.
Yeah.
I'm here again.
Okay. Okay.
Sorry, it's the network. Okay, so what I was saying, that the most beautiful thing is that you really look behind the rationale of why the refractive index is different because of the cortex of the cell and so on, and the work of the brain I found very, very interesting and really very practical and very important for the surgeons. And I was wondering, in these moments that we live with the virus, did you think in an application of identification of or detection of the COVID with this type of sensors?
Jose Luis.
I mean, we could try. We could try. We have samples here in the hospital, but now I think there are very accurate techniques to identify the virus. There is some molecular biology, this PCR technique that everybody knows about, or even the antigen test. I mean, there are many techniques, very extremely accurate, to identify the presence of the virus, and also to identify not only the presence of the virus, but also the variant of the virus. And it will be really difficult with a system like our biosensor to identify the different variants of the virus, the different mutants of the virus. and we know that by sequencing the virus. But as I say, I mean, we could work on that, but I don't know if it's very useful because there are a lot of techniques very accurate for this reason.
But those techniques sometimes they require a lot of time, and if you can develop kind of a sensor.
Not really. I mean, an antigen test takes minutes, and a PCR takes maybe an hour, 2 hours. I mean, there are very fast techniques to identify the presence. If you want to identify the variant, the mutant, it takes more time because you have to sequence the virus. But this is just more specific.
And this is always thinking in the detection on the body or so on. But do you think it's possible to perform the detection in air, which is where it propagates and it will alarm if instead of just a simple sensor of CO2, having a sensor that able to detect, okay, in this room there is a high concentration of the virus?
I don't have a clear answer to this. I think it would be very difficult. I mean, to manage virus is not really easy. I mean, we can make these biodosimeters with bacterial, with living cells, with genetic material from virus, but not with entire virus, with the whole organism. I mean, and even to detect this virus with a system like our biosensor, I don't know. I don't really know. But I guess it would be really difficult. It would be a challenge, challenging application of the biosensor.
Yeah. Yes.
I think we would need special installations to work with that virus. Otherwise, it's impossible. You need
safety, very high.
very safety place, approved place, which are special conditions to work with this very dangerous virus or like other virus. I think in Spain there are only 2 or 3. Or how many
labs?
Yeah. I mean, work in experimental conditions for working with living virus. I mean, it's really complicated. You have a special room, special wide room with special conditions, and there are very few in Spain that can allow to work with virus, with this type of virus, which are really aggressive and very dangerous. So we are not in this situation, of course.
Okay. So thank you very much for your answers, and congratulations again to the company and to the work that you are performing.
Thank you.
Yeah, thank you.
Anyone else would like to ask something? Okay, so maybe we can close the section. I would like to thank again the presenters for coming virtually here today and explaining their fantastic work. It's been super interesting. Thank you very much. And okay, we clap and thank you.
Thank you.
Thank you. Thank you.
Thank you.
Thank you.